Hello, good morning, and welcome everyone to another Rapid Novar monthly webinar. My name is Wei, and will be your host and moderator for today's presentation titled Rational Antibody Design and Engineering with Next Generation Protein Sequencing. Towards the end of the presentation, we will be doing a Q&A, so if you have any questions, we welcome you to type it into the Q&A chat below. As well, the presentation is being recorded and will be made available to everyone shortly afterwards. Next slide, please. So in today's webinar, we will have a keynote presentation from Dr. Jennifer DeLuca from Colorado State University, who will be talking about creating a recombinant antibody palette to study mitotic cell division. Afterwards, our scientific sales executive, Dylan, will provide an overview of Rapid Novar's next generation protein sequencing technologies for the de novo sequencing of monoclonal and polyclonal antibodies. Next slide. A quick introduction of our speakers today. Dr. Jennifer DeLuca is a professor and associate chair of graduate studies at the Department of Biochemistry and Molecular Biology at Colorado State University. Using a wide array of bioanalytical approaches, the DeLuca lab investigates the association between chromosome missegregation and cancer and how key phosphorylation events on mitotic proteins regulate the chromosome segregation process. Dr. DeLuca is also the recipient of the 2021 American Society for Cell Biology Lifetime Achievement Fellow Award. Thank you so much for joining us today and for speaking as well. Next slide. Dylan is our scientific sales executive covering the North American West Coast. He graduated from the University of Toronto with a master's in laboratory medicine and pathobiology. Dylan is very passionate about science and new technologies, especially with regards to accelerating research through the use of next generation protein sequencing. So without further ado, I'll turn it over to you, Dr. DeLuca, and thank you so much.
Okay, great. All right, so thanks very much for the introduction and for the invitation to participate in this webinar series. Again, I'm Jennifer DeLuca, and our lab at Colorado State University studies mitotic cell division. And I'll start off with a very brief description of our lab's research for just a few minutes, but then really the focus of today's talk is going to be on how we are developing and using recombinant antibodies and antibody derivatives to help move our research on mitosis forward. Okay. All right, so again, just some very quick basics. Mitosis is the process by which a single cell divides into two identical daughter cells. And the goal of mitosis is that the two resulting daughter cells receive an exact copy of the original genetic material, which is packaged into chromosomes. And it's critical that this happens exactly correctly and exactly evenly every time. And so the reason that we care, and many people care, so much about the fidelity of chromosome segregation is that when errors do occur in this process, the result can be the formation of aneuploid cells. So here an animation. And so aneuploid cells, this is a state in which the resulting daughter cells have either too many or too few chromosomes. And the reason that we're concerned about this is that this can lead to dire consequences for human health, as aneuploidy has been known to be intimately linked to both tumor initiation and progression and actually other human pathologies. And so that's illustrated here in these karyotype analyses of healthy cells. These are bladder cells. Healthy cells, which have two copies of each chromosome, everything looks great, just like it should. And then we have an example here of two bladder cancer cells, which hopefully you can see are wildly aneuploid with multiple copies of some chromosomes and either 0 or 1 copy of other chromosomes. And so essentially our lab tries to understand the mechanisms that cells use to end up like this with a normal karyotype and not like this, and to avoid these pathologies. Okay, so again, just a quick reminder on the slide is that during mitosis, the duplicated chromosomes line up in the middle of the cell. You probably remember this from 8th grade biology. So these chromosomes line up in this formation called a metaphase plate. And once all the chromosomes are properly aligned, the sister, let's see if I have an animation, the sister chromatids are then pulled apart to the opposite sides of the cell, and the cell will then eventually pinch in two, hopefully resulting in two identical daughter cells. And I'm going to play this movie here, and hopefully you can see that in a real living cell, where once the nuclear envelope breaks down, the chromosomes find their way to that metaphase plate. We'll show it again. And once this last chromosome joins in and becomes correctly attached, the chromosomes can split apart, and then the cell divides into two. Let's see. Okay. All right, so in order for these chromosomes to line up at the metaphase plate and then to be pulled apart in anaphase, the absolute key event that has to happen in all mitotic cells is that these chromosomes must attach to spindle fibers of the mitotic spindle called microtubules. So these are dynamic polymers that grow and shorten and grow and shorten, and they're made up of the tubulin subunit. And so the region on the chromosome where these microtubules are going to attach is this very large protein structure called the kinetochore. And this is really the main focus of our lab, is this structure called the kinetochore. So these microtubules are going to plug into the kinetochore. And as they grow and shorten and grow and shorten, they're going to push and pull these chromosomes around the mitotic cell to eventually align them at the metaphase plate at the cell equator. And hopefully you can see that in this movie, where the microtubules are shown in red and the kinetochores in this case are shown in green. And hopefully what you're seeing is as the microtubules are growing and shortening and growing and shortening, they're pushing and pulling those chromosomes around the cell. Okay, so the protein, let's see here. So the protein that physically makes this linkage, that links kinetochores to microtubules, is called HEC1. And so again, the kinetochore is made up of hundreds and hundreds of proteins. We won't get into any of those, but really the key protein that our lab is really concerned with is a protein called HEC1, which again physically connects to these spindle microtubules and really allows these chromosomes to line up and then be pulled apart. And much of our lab's research focuses on this attachment between kinetochores and microtubules through this protein HEC1. All right, so this is my last of my background slides, and I just want to mention that kind of our current research questions are all kind of involving this kinetochore microtubule attachment process. So we're interested in understanding how this protein HEC1 binds to microtubules and generates force for chromosome movement. We're interested in how the attachment strength at that attachment point is regulated so that if things go wrong and there are errors in attachment, those errors can be corrected. We're interested in understanding how cells know when all the kinetochores are attached to microtubules, so the cell can be signaled for mitotic exit. And then finally, a big push in our research is trying to understand how oncogenic stress in cancer cells leads to defects in kinetochore microtubule attachment. And so these are all the projects, the biological projects that our lab is working on. And this is where I kind of segue into why we're here today, which is that all of our projects, hands down, rely heavily on antibodies and antibody reagents. And so, excuse me, so just as kind of an example, I would say 90% of the experiments that we do in our lab involve these three techniques. These are, and they're all antibody based. So immunofluorescence techniques, where cells are immunostained with various antibodies and then imaged on the microscope. And so here we just have a panel of mitotic cells. And I just want to point out here, the chromosomes are in red, the microtubules are in white, and here all these little paired dots are the kinetochores. And so much of our research uses immunofluorescence. We also use Western blotting very routinely, which is an antibody-based technique, as well as immunoprecipitation, again, antibody-based technique, where we carry out, for most of our projects, immunoprecipitation and mass spec analysis, usually to identify interacting proteins. Okay, so how, being a mitosis lab that doesn't really think too much about antibody development or technology, how did we get involved with this whole process of generating and diversifying recombinant antibodies? And again, for this audience, this probably doesn't need to be made clear, but just to define what I'm talking about, recombinant antibodies are reagents that are made in vitro by cloning, expression, and purification of the antibody heavy and light chains from cell culture, meaning they're made in the lab, not in an animal. So the question again, how did we get here? Why are we interested in generating these recombinant antibodies? Okay, so as with most cell and molecular biology research labs, we and many, many others constantly grapple with the disadvantages of traditional commercially available antibodies. And so just a couple of those examples are listed up here. So these antibodies can often exhibit poor reproducibility due to lack of standardized reagents. So as many of us probably know, there's you order an antibody from a company and there can be high variability from lot to lot. And sometimes the antibodies can just stop working altogether. And so this is really not good for reproducibility in your research. Second, there's no guarantee of continued availability of these antibodies. These traditionally generated antibodies require active maintenance or continued production in animals, and this can naturally lead to loss of the product or discontinuation. Finally, and this is a big one, or thirdly, this is a big one, antibody companies sell their antibodies at very high cost typically, and in some cases, depending on the type of experiment that you want to do, for example, if you're trying to label an antibody or generate a super resolution probe where you need a lot of antibody to start with, these high costs can be prohibitive, again, depending on the experiment. And then finally, the use of a large number of animals in these traditionally generated antibodies can raise ethical concerns. Okay. All right. So, these problems have been around forever, a very long time, but what pushed us over the edge to go into this foray of recombinant antibodies? So the answer is very specific. So what pushed us over the edge was the abrupt loss of a commercially available high-quality monoclonal antibody to our beloved kinetochore protein HEC1. So for years and years and years, there's been a commercially available antibody called 9G3 to HEC1. It's a monoclonal antibody that's been available since about 1998. It's a gorgeous antibody. It's a beautiful antibody. It's used by probably hundreds of labs, not only labs studying mitosis and chromosome segregation, but it's used as a very standard marker for specific stage of the cell cycle, mitosis. And so many, many labs use this antibody, and it's been very reliable, and it's just been a really nice antibody. Then in about 2018, kind of bizarrely, the antibody just stopped working from commercial companies. So we would order different lots, we would order it from different companies, and it just, became crappy antibody. It went from a great antibody to just terrible. And what was really interesting around that same time, I was receiving emails from lots of people around the country, around the world, kind of commiserating about the same problem. And so here are a couple of those example emails. Basically, hello, I understand there have been global issues with the 9G3 monoclonal antibodies. What's your opinion? Dear Jennifer, we have been having trouble with the commercial 9G3 antibody. New vials are not working. Do you have any experience with this, or how are you solving this problem? So not only were we experiencing this, but it was kind of global. And so really there was kind of a panic in the mitosis field that this really reliable, useful antibody was just gone forever. Okay, so about the same time, this whole saga was unfolding, I was discussing this problem with a colleague in our department, Tim Stasevich, who uses antibodies, antibody fragments and nanobodies to study various processes in living cells, including transcription and translation. And so Tim just kind of casually suggested that we try to sequence the antibody and then use that sequence to generate a recombinant version on our own. And we had not even really, this was not on our radar. We didn't even know this was really a possibility, but we thought this sounds perfect, and we thought we'd give it a try. So we set out to obtain the primary sequence of the antibody and then attempt to clone it and purify a recombinant version. So as we got deeper into this project, we realized that this approach potentially had a large number of advantages and could potentially solve a lot of our antibody-related problems that I listed out at the beginning. So for example, we would be able to immortalize the antibody, and once the sequence is in our hands, then we can generate this antibody in perpetuity. Our grandkids can generate the same antibody. In addition, this would allow us to have increased antibody reproducibility. The antibodies are generated from an invariant sequence, and so the reproducibility is kind of baked in. And a kind of a big advantage for us would be that we could generate high yields of antibodies in-house in our own lab, which is important, again, if we're doing experiments that require these large amounts of antibodies. And then probably most exciting for us was that we realized that once we had the sequence in hand, we could increase our reagent versatility, and this would create new opportunities for unthought of experiments. So for example, we realized that once the sequence was in hand, we could change the species specificity of the antibody. We could generate all sorts of types of antibody fragments, And importantly, we could generate genetically encoded fragments for tracking antigens in living cells. Okay. All right, so enter Rapid Novar. So as we kind of were looking around, trying to figure out how to do this, how to get a sequence for an antibody, Rapid Novar seemed like the kind of premier company that was providing the service. So Rapid Novar, according to their website at the time, offered monoclonal antibody sequencing services. And, this is just information from their website, which looked great to us. It required relatively small amount of antibody, 100 micrograms of purified antibody. They would carry out mass spec-based sequencing. It works on all types of antibody types and species. What we would get back out of it would be a report on the heavy and light chain, the full sequences that are annotated to indicate all the different domains, the constant regions, the variable regions, the hypervariable regions, and the turnaround was about 14 days. So we were excited and we thought we'd give it a try. So we did. We submitted our HEC1 9G3 antibody, or at least what we had left of it, to Rapid Novar for sequencing. Okay, so about 2 weeks later, we received kind of a soft, an electronic copy of the sequences for both the heavy and light chains. And so importantly, these included the annotations for the various domains, which are shown here. The heavy chain constant regions and light chain constant regions were annotated. The variable domains from the heavy chain and light chain were annotated. And it also included within the variable domain, the hypervariable regions, or the CDRs. And so that's going to be important later. And so essentially we get this fully annotated sequence of the heavy chain and light chain in hand as a soft copy. So we're now ready to, with the sequence in hand, we could begin generating our recombinant version of the HEC1 antibody and hope it's successful. Okay, so the first thing that we did was to clone both the heavy and light chain sequences into expression vectors. And for this 9G3 antibody, we initially took the amino acid sequences of the entire heavy chain and entire light chain, and then reverse translated the protein sequence back into DNA sequence. And importantly, because we knew that we were going to eventually express these vectors in human cell cultures, we made sure that we optimized the codon usage in the DNA sequence for expression in human cells. And we did this through the company that we ordered the gene blocks from, which is GeneArt. Okay, so once we did this, we order our gene blocks containing our codon optimized DNA encoding for the entire heavy and light chains. All right, and I also just want to point out here that as a note is that we had to make sure that we included what's called a signal peptide at the end terminus of both the heavy and light chains. So these signal peptides are short stretches of amino acids that direct the newly made antibody or any protein to be secreted out of a cell. And in our case, it will secrete the antibody out into the cell media. And so I just want to bring this up because it's important to note, because the sequences from purified antibodies will not contain the signal peptides because they're naturally cleaved off of the antibody, so they won't show up in the final sequence. Okay, so finally, once the gene blocks are ordered and delivered, we can then simply clone the DNA from the gene blocks into our expression vectors of choice. And we happen to use a CMV-driven expression vector to ensure high levels of expression. Okay, so step one, we've gotten the sequence and we've cloned the heavy and light chains into our expression vectors. So now we move on to the next step, which is to transfect the plasmids into our cell cultures. So we now have our expression vectors, one heavy chain, one light chain, and we're now going to transfect. So in our lab, there are lots of options that we found for kind of the cells that you can transfect your plasmids into. And we've tried a few different ones, and we've had the best luck with a HEC293 cell line that grows in suspension. And these are called XBY 293F, and these are specifically formulated so they can grow at high density in suspension. So we've really stuck on using these XPI 293 cells. Okay, in addition, we actually found a very low-cost transfection reagent because we're going to be transfecting into, medium-sized cultures of about 30 mils. And so we're trying to lower our cost with the transfection reagent. And so we found through several companies that you can order this PEI at relatively low cost. Okay, so now next step is to transfect our heavy chain and light chain plasmids at a ratio of 2 to 3 into our XPI cells. And then very simply, the next step is to wait 5 days for optimal expression and then secretion into the media. All right, so we've made our clones, we've transfected into the cells, and now we wait for the cells to produce the antibody and then secrete into the cell media. All right, and then the final step of the process here is to purify the antibody. And so for this, we simply take our cell cultures, we centrifuge the cells, we pellet the cells, and then shown here, the cells pellet at the bottom of the tube, and then we can simply collect our supernatant or the cell media, which contains the secreted antibody. We then run the supernatant over a protein A cephalose column. And again, as most of you probably know, protein A will directly bind to the FC region of IgG antibodies and some other antibody classes. So the antibody is then retained on the protein A cephalose column. So we then simply wash with our column buffer to remove any nonspecific binding, and then we elute with a low pH buffer. So here comes our antibody off the column, and we simply neutralize the eluate, which contains the antibody with a basic buffer, and then we can now aliquot and freeze down our samples. Okay, so for our first try, for again, trying to generate this or regenerate this HEC1 antibody by producing a recombinant antibody, we started with a 30 mil cell culture, and we were able to, again, on our first try, get a yield of about 1.1 milligrams of antibody. And so here on the right, I've just shown a Kumasi stain gel of the purified antibody. This is just, the lanes here are just decreasing concentrations of the purified antibody. And hopefully what you can see in the gel are the heavy chains here, which are both the glycosylated and the non-glycosylated heavy chains. And then down here we have our light chain. So it's a really, and up here, we just have some non-denatured antibody that didn't get denatured on the gel. So we have a really nice clean prep of antibody. And I also, even though I haven't talked about any of these yet, I just wanted to kind of put up this chart, which shows, a wide swath of some of the antibodies that we've actually now made in a recombinant form, just to show you the kind of the range of yields that we can get from these 30 mil cell cultures. So we don't really know the source of the variability yet, but there is quite a bit of variability, but the yields do range from anywhere from quite low to about 100 micrograms to very high with 2 milligrams, which, according to, buying this from a company would be probably $10,000. And once we have the sequence in hand, we can produce these antibodies for, under $100. Okay. So that's kind of the general gist of producing the antibody from the sequence that we had gotten from Rapid Novar. And then the next step that we needed to do would be to validate this new recombinant version of our HEC1 antibody to make sure it actually works. And so we did this through both immunofluorescence and Western blotting. These are just the standard techniques we use in our lab. So here we have mitotic cells in various phases of mitosis. And HEC1 is a kinetochore protein, so it should be recognized as these paired dots in these mitotic cells. And so what you see hopefully here with our recombinant HEC1 antibody, we see gorgeous staining, we see these nice paired dots, high signal to noise, very low background, and we see just really kind of perfect kinetochore staining here. And we were also able to show this antibody was specific to HEC1. We depleted HEC1 using siRNA techniques, and you can see there's no more immunoreactivity here. We did the exact same validation with Western blotting. where in the left lanes, you can see control HeLa cell lysates. And then in the right lanes, we have lysates from HeLa cells depleted of HEC1. And these are just 3 different concentrations of lysate that we tried. And hopefully what you can see is that the new recombinant HEC1 antibody really nicely recognizes this 73 kilodalton band, which corresponds to HEC1 with very little background staining. And then when we deplete HEC1, the staining is completely gone. So we were really just delighted with our new recombinant antibody. So then I just want to show you this slide where we just kind of for fun, we wanted to compare our recombinant, new recombinant monoclonal antibody to our best commercial 9G3 that was left in our freezer. And here, if you look at the yellow arrows, we're just pointing at a metaphase cell stained with the new recombinant HEC1 antibody. You can see again, these gorgeous paired dots with very low background. The kinetochore staining is really perfect. And then in the metaphase cell stained with the commercial 9G3 antibody, which had begun to lose quality, we can see it's kind of a mess. We have high background. Not only do we have very little kinetochore staining, but we also see the entire spindle is lit up with the antibody. So it was just really terrible. So kind of in summary from this experiment, we were really delighted to have gotten the sequence and then successfully generated our 9G3 recombinant antibody. So we were so excited that we went on and did a few more. So we ordered from Rapid Novar, we obtained sequences for 2 more of our kinetochore antibodies, antibodies to our kinetochore proteins. These include a very common kinetochore marker called SEMC, which again worked beautifully. And then I just want to point out on the left, we actually made a recombinant version of an antibody to KNO1 that's specifically phosphorylated. So as was mentioned in the introduction, many of these kinetochore proteins depend on phosphorylation for their regulation. And so what we wanted to do was to try to produce a recombinant version of a phosphospecific antibody, kind of just to see if it would work. And it worked beautifully. Our KNO1 phosphoantibody recognizes kinetochores as it should in early mitosis. And what we found was that the phosphorylation on this antibody, or sorry, the phosphorylation on this protein precipitously drops as cells move into metaphase, which was really exciting. We went on to validate this antibody to show that it was phosphospecific by running these on a gel, staining with the antibody in the presence and absence of a phosphatase, which you can see here, hopefully in this Western blot, that when we treat with the phosphatase, we lose the immunoreactivity. So we were really excited and really happy with what we had gotten from Rapid Novar, which were these sequences which we could very successfully turn into recombinant antibodies. Okay. All right. Okay. So given the success that we have with these first antibodies, we really wanted to keep going and generate more useful recombinant antibodies. But I'll have to say at this point, given our funds, we wanted to investigate some lower cost options. And so for this, we decided to try to obtain sequences from monoclonal hybridoma cell lines rather than from the purified antibody. And so I won't go into a lot of detail, but in this approach, it involves whole transcriptome sequencing of the mRNA that's isolated from the hybridoma cell lines. And so for this sequencing procedure, the companies typically require about 1 million to 1.2 million cells, which is about 135 millimeter small dish. And so you send in the cells, and what you get back, similar to Rapid Novar, would be annotated heavy chain and light chain sequences. So the advantages of doing it this way is that the cost is actually significantly less than sequencing the purified samples. We can get sequencing from various companies from about $800 to $1,200, versus anywhere from $5,000 to $14,000 for the purified samples. The disadvantage, however, is that you actually need the hybridoma cell line, which is not, which at least for our lab, we don't make a lot of our own hybridomas, so we have none. And so you would need to obtain the hybridoma cell lines. You could either make them yourself, which again goes back into high cost, or what we did, we phoned friends. So we asked our buddies who did have these hybridoma cell lines if we could have some small samples to do the sequencing. And so we're very thankful to our friends Jakob Nilsson and Gary Gorbsky for providing a couple of these hybridoma cell lines. Okay, so again, I'm not going to go through the whole procedure again. It's exactly as the same as I described before. You obtain the sequences from the hybridoma lines. You then go through the cloning, the transfection, and the purification, just as I described before. And again, using this technique, we were also able to generate some very nice recombinant antibodies. And again, I'm not going to go into detail on all the antibodies we tried this way, but a couple are shown here, where we generated a recombinant antibody to a very nice kinetochore checkpoint protein called BubR1, which localizes to kinetochores very beautifully and recognizes the band by Western blot. But then what I really want to point out is that we made a recombinant antibody to a conformation-specific version of a protein. So what do I mean by that? So this protein MAD2, which is also a kinetochore protein, and it's involved in checkpoint signaling. It can exist in two forms, active and inactive, and the active form is open, and the active form is closed, and the inactive form is open. And so what we did was we actually made this antibody specifically to the closed active form. So now we were successful in generating this recombinant MAD2 antibody, but it's a conformation-specific antibody that only recognizes a specific version of the protein, which is going to be important in just a few minutes in the talk. Okay, so I just want to give kind of a summary so far, and then I'll get into kind of how we've diversified these antibodies from the sequences. Okay. So summary so far. So we obtained sequences for monoclonal antibodies, both through purified samples and through hybridoma cell lines. And we were able to generate new recombinant versions of these antibodies that really work great. So I kind of wanted to go through how we've done this and how we obtain the sequences and maybe some of the kind of overall the advantages and disadvantages of doing them in different ways. Okay, so we've sequenced purified antibodies from Rapid Novar. The advantages here are there are a huge number of purified antibodies available that you could do this for. So really any antibody you want, any monoclonal antibody you want, this is an option. And we were really lucky to have 100% success rate working with the company. The disadvantages are right now, the costs are quite high. For most biomedical research labs that run on NIH funding or NSF funding, in some cases, these can be prohibitively high. The new customer price is $5,000, which is somewhat reasonable. And then the regular price for the antibodies go up to about $15,000. And so there are options for people. They could write grants specifically to sequence antibodies that could kind of help with this price. And then I'll also point out that the services are routinely available for monoclonal antibodies, but the technology, at least as far as I know, which we may hear from Dylan, this has changed, that the technology is not quite there for polyclonal antibodies. And while I think these are techniques that are in development, and maybe there are some options for sequencing polyclonal antibodies that are successful, I think the costs for doing that through a company are still quite high. And really for a small research lab, prohibitively high. And so the hope is that companies like Rapid Novar are going to bring down these prices or get these technologies moving and then get these prices down. So kind of most labs will have access to these reagents. Okay, so the second approach that we've used is this whole transcriptome sequencing from the hybridoma cell lines. And again, the big advantage here is that they're relatively low cost. Once you have the hybridoma cell line in hand, you can get them sequenced for about $1,000. However, the disadvantages are that you need the hybridoma cell line in hand. And of course, a similar service is not yet available for polyclonal antibodies. And then finally, and I haven't really talked about this yet, but I will in just a minute, is that a third option is that you can actually obtain sequences from publicly available databases. So if any antibodies have been sequenced, if any antibodies have been crystallized and the atomic structure has been solved, you can actually get the sequence from the atomic structure. And then there are some services and databases that will publish kind of known antibody sequences. Advantages of this are they are absolutely free. You just have to kind of troll the internet to find them. And the disadvantages is right now, there's a limited number of sequences. So, for us, when we try to find sequences for kinetochore proteins, they're really just a handful out there, and it's just not kind of widely available yet. Okay, so this is the last part of the talk, the last maybe 10 minutes. So now that we have the sequences in hand and our recombinant antibodies have been successful, what can we do with them? How can we increase our reagent versatility and diversify these very cool reagents? And so the answer is that yes, we can do this. And we've had a lot of fun kind of messing around and trying different approaches. And for the rest of the talk, I'd like to tell you about 3 things that we've done to increase kind of or diversify our antibodies. One is that we've been able to swap the species specificity for different antibodies. The second is that we've been able to produce full-length bivalent antibodies from freely available fragment sequences online. And then finally, we've been able to generate antibody fragments and genetically encoded fragments, such as scFvs, from the sequences that we obtained from, for example, Rapid Novar. Okay, so the first idea that we had that we wanted to try out was to try species swapping. And so this is essentially taking a mouse antibody and then using cloning to turn it into either a rabbit antibody or a human antibody or a chicken, rat, whatever a species of choice. And the reason why we had a very specific reason for doing this, which is that many of our cell biology experiments, especially in direct immunofluorescence, are limited by the antibody species that we have. And that's because a fluorescently labeled secondary antibody binds the constant domains of the primary antibody, which binds the target. And importantly, the secondary antibody specifically recognizes the primary antibody constant regions from a particular species. So all that just means that it limits the combination of antigen targets. And essentially, you can't co-stain 2 antigens with 2 mouse antibodies or 2 rabbit antibodies. You have to make sure you have one of each so that you can do a co-staining experiment. Okay, and this comes up all the time in our lab. We'll have a great monoclonal mouse antibody that we want to co-stain, but the only other antibody you have is another mouse monoclonal antibody. And so it really does limit what we can do. So now that we have the sequences in hand, we thought maybe we can solve this problem. And so we did this by trying to generate a new HEC1 9G3 primary antibody that is now recognized instead of a mouse secondary, a rabbit secondary. And so the way we did this was simply to combine the variable domains of our HEC1 mouse antibody with the constant domains from a rabbit antibody. And then now we created this kind of new hybrid, which is a rabbit HEC1 antibody. And so we did this. It was pretty simple, just do some cloning. And then the antibody turned out great. And so if we look at this top row, what you can see is that here's a mitotic cell in a metaphase plate formation. And now this is our new rabbit HEC1 antibody stained with a rabbit secondary. You can see these, paired dot kinetochores. They look really good. And then now if we take the same sample and we stain with a mouse secondary, it no longer is recognized by the mouse secondary. And now we've been able to do this for all of our antibodies. We've been able to basically make human, mouse, rabbit, rat, chicken, whatever we want from any of our recombinant antibodies. And so here's an example where we took, again, our HEC1 antibody, our original HEC1 antibody, and then we've now kind of converted it into a human anti-HEC1 antibody that's recognized by human secondary antibodies, but no longer mouse. Okay, so, I'm getting close. All right, so the second idea that we had using these recombinant antibody sequences was to produce full-length bivalent antibodies from an scFv or a single chain variable fragment sequence. So I just kind of take a segue here to kind of remind everybody that right now what's happening is there are relatively new in vitro methods that are in process of being developed to produce scFvs in the test tube. So for example, phage display screening is a way to kind of test for and screen for a new scFv, again, without using an animal, with just starting with a target sequence. So because of this, there are growing numbers of published scFv sequences freely available online. So we had the idea to kind of troll, again, the internet for these published scFv sequences, take the sequences and then reconvert them or reverse engineer them back into a full-length bivalent antibody that we can use for immunofluorescence immunofluorescence, Western blotting, immunoprecipitation, et cetera. And so it's, again, very simple. We simply order the gene blocks for the heavy chain and light chain variable regions of the sequenced scFv. And then we combined them with our constant regions of our mouse, rabbit, human, whatever, IgGs, to produce this brand new full-length bivalent antibody. And we've now done this for about 4 sequences so far. And I want to show you kind of our most successful one, which is our tubulin antibody. So we were able to obtain the sequence of a tubulin scFv from this database called the Antibodies Chemically Defined database. So we obtained the sequence, we ordered gene blocks corresponding to the heavy and light chains, we cloned these onto our various scaffolds, and then we were able, again, to produce these really nice bivalent full-length tubulin antibodies, which really rivals our best tubulin antibodies that we can buy commercially. So here, this one happens to be the rabbit tubulin antibody. You can see the microtubules very nicely stained to the mitotic spindle. And then if we look here at the Western blotting, we can see that this new full-length tubulin antibody really made de novo from this scFv sequence, recognizes this is a whole cell helolysate, beautifully recognizes this 55 kilodalton band corresponding to tubulin with very little background. And so we have done this again for several different antibodies, and we're continuing to mine the literature for relevant sequences to produce more of these reagents. Okay. And then the final thing I want to mention is that in terms of diversification of these reagents, is that we've been able to generate from the primary sequence antibody fragments, many types of antibody fragments. And what we're most excited about are the genetically encoded scFvs. So again, these are single chain variable fragments. So antibody fragments, again, as most of you very likely know, are smaller versions of full-length antibodies. So why? Why would you want to use a fragment versus a full antibody? So I apologize for all the text on the slide, but I wanted to make sure I hit all these points. And so a smaller size allows better penetration of tissue samples. It's better to identify buried epitopes. These smaller fragments can bind targets without inducing cross-linking, which reduces steric effects, which could introduce artifacts into your experiments. They're especially important for use in super resolution microscopy, where the resolution that you can obtain directly depends on the size of the antibody probe. So the smaller you can get your probe, the higher resolution you can get for your structures. Okay, and then for us, I think the most important thing is that we can use these scFvs, or small fragments. We can fuse them to a fluorescent protein, genetically encode them for expression in living cells, so we can track our antigens in real time. So why is this important? This feature is especially essential when your antibody recognizes not just a protein or an epitope, but a post-translational modification, or, for example, a specific protein conformation, because these cannot be tracked simply by expressing a GFP-tagged version of your protein. So there are really limited ways of tracking protein modifications and protein conformations. And if we can generate genetically encoded GFP-tagged scFvs, this would completely solve that problem and allow us to track these types of modifications and conformations. And again, as I mentioned before, in mitosis, kinetochores are heavily regulated by phosphorylation events. So it would be terrific for our group to be able to track these phosphorylation events in real time. Okay, so that's why we want to do them. So how do we do it? So again, once we have the sequence in hand, for example, from Rapid Novar, we can simply take that sequence and then generate our native scFv. And so the way we do this is that we basically extract the variable domains from the heavy chain and light chain, and then we can connect them with a flexible linker, which hopefully is shown here. So we have our variable region from the heavy chain, variable region from the light chain, connected with a flexible linker. We can then add on the sequence for GFP, so that when this is expressed, it's going to produce a green fluorescent protein. Okay, so we did this for one of our recombinant antibodies, and the one we wanted to try first, again, was a phosphoepitope, because we really wanted to try to track phosphorylation in living cells. So we generated our phospho-KNL1 scFv tagged with GFP. We then expressed it in our HeLa cells, and we did live cell imaging to track, again, this real-time phosphorylation. And so first, I'm going to show you the stills here, where you can see in these living cells, we have these little dots are recognizing the phosphorylated form of this kinetochore protein. And then as the cells progress through mitosis, what you see is the phosphorylation precipitously drops. And then you can see at the metaphase plate, just a few kinetochores retain this phosphorylation. And so again, this was really exciting for us. Here's the movie. And again, hopefully what you'll see when it plays again is that at the beginning of the movie, as mitosis begins, this phosphorylation on this protein is very high with these dots everywhere. And then as the chromosomes line up at the metaphase plate, you can see that this phosphorylation gradually decreases until it's completely gone. And only once this phosphorylation is gone, the chromosomes can split apart in anaphase. So we're super stoked because now we can test the parameters that lead to KNO1 phosphorylation and dephosphorylation during mitosis in living cells. Okay, and I will point out, we've tried this for a few of our other sequenced antibodies, and we've had success with our conformationally closed version of the mitotic checkpoint protein MAD2 and made a really nice genetically encoded scFv to that. But aside from that, I will also say that a challenge that we've run into is that the generation of these scFvs from the native sequence has actually been we haven't had a great hit rate. And so it's only been successful in about 30% of cases. So that just means if we take the native sequence that we get from the company and we take the variable regions, link them together and express them in cells, we only have about a 30% hit rate. So we're trying right now to optimize the success rate for generating scFvs from our sequences, and by doing a couple things. And so what we're currently working on is a scaffolding approach, where we first identify the hypervariable domains within the sequence and then graft these onto known or various framework domains within the variable region, which we're calling the scaffold. And so what we're doing is we're trying to go through the literature and find native scFvs that have worked and use the scaffolds or the framework regions within the variable domains that have shown success, and then combine those with our extracted hypervariable domains to try to generate a successful scFv. And then the other thing we're trying to do, which is kind of brand new, is we're screening through different combinations of hypervariable domains and framework domains, first by using AlphaFold structural predictions. And so in this way, what we're trying to do before we go through all the cloning and building and testing, we're running these through AlphaFold predictions to try to identify the most stably predicted structures to give us an idea of which of the scVs might work in cells. Okay, all right, and that is about it. So my closing points are that we've developed methods and reagents to produce high yield, relatively low cost antibodies and antibody fragments in our own lab. Our current area of focus is that we're trying to, as much as we can, gather hybridomas and published sequences to increase the number of available recombinant antibodies for folks to use. And we're also trying to improve our pipeline to generate genetically encoded scFvs, and especially to protein modifications and protein conformations, because we think these will be very valuable reagents for the field. And then finally, just a couple of the current challenges are that there are high upfront costs if you don't have the sequence or the hybridoma cell line. And again, many antibodies produced by labs are polyclonal. And so sequencing these polyclonal antibodies may be cost prohibitive, but hopefully the technologies are being developed that sequencing polyclonal antibodies will come on the scene soon. And with that, I just want to thank the people who did this work in my lab. Jean Mick and Keith DeLuca really spearheaded this entire project. Jean is our cloning guru and has done all of the cloning for all these projects. And then Keith takes these clones, expresses them, purifies the antibodies, and does all the downstream validation. I also want to thank two members of our departments, Tim Stasevich and Ning Zhao, who really have helped us on the kind of the Frankenbodies and trying to test different scaffolds for our scFvs. And then finally, Jakob and Gary provided hybridoma cell lines for proof of concept for generating recombinant antibodies using this technique. Okay, and with that, I am done. And I will stop sharing. Okay. All right. Hopefully, okay, someone's still there. Okay, all right.
Yeah, I'm still here. All right, I'll start sharing my screen now.
Okay.
Okay. Right. All right. Okay, so first, just wanted to say thank you, Dr. DeLuca, for the great presentation. Really appreciate the insights you provided with regards to the benefits of recombinant antibodies and how having the sequences can also open additional avenues and increase reagent versatility. Also want to just give a quick reminder to everybody that we are going to be doing a Q&A session after this. So if you have any questions, either for Dr. DeLuca or myself, feel free to just toss that into the Q&A section. We'll get to those at the end of the presentation. All right, so for my part of the talk, I know that Dr. DeLuca already touched on it a bit in her presentation, but I'd like to now focus in on really what our capabilities are here at Rapid Novar and share more about how we're actually able to get the sequences of antibodies directly from the protein itself and how this may be beneficial for those in the antibody space and as well as other researchers in additional fields as well. So really, I think the logical starting point for us is to share a little more about the methods that we use when performing our de novo protein sequencing. Our core capabilities really are a combination of our in-house wet lab and bioinformatics teams, and then pairing this with our proprietary mass spec-based technology and software. So here we've got sort of a typical workflow for our monoclonal or mAbs sequencing. So we actually will start with a purified sample, which is then digested with a number of enzymes before being flown on the mass spec. And then our algorithm is able to take this data from the mass spec and assemble it into full-length heavy and light chain pairs. And then the sequences are checked and double-checked by our bioinformatics team before being sent off in a nice cohesive report. So I know when it's condensed into sort of a little diagram like this, makes it seem quite straightforward, but there really is a lot of work going on behind the scenes as well. So now you may be wondering how confident we can be in the sequences that we generate. And I know one of the common issues that is regularly brought up is how do we differentiate between the isobaric residues, leucine and isoleucine? So for this, we actually use our wild technology, which is a method our team was the first to commercialize. And using this method, we're able to see a 43 Dalton shift in leucine compared to a 29 Dalton mass shift in isoleucine. And this really gives us the capability of getting 100% confirmation for every possible IL site that we're looking at. Now, outside of the IL determination for the sequence as a whole, we set really high standards for what we're looking for from a coverage standpoint, as well as what we're looking at for overlapping peptides. So here is what a typical coverage view might look like for a monoclonal antibody chain. So as you can see, we get great coverage here along each of the amino acids, and each one of these individual colored bars represents a unique peptide. And each of the different colors corresponds to a different enzyme that was used during the digestion. So these points in combination with our rigorous checks and great bioinformatics team allow us to really provide a full-length sequences with 100% accuracy, which I'm happy to hear that Dr. DeLuca has also had a 100% success rate with all of our projects that we've worked on together. So what's also great about our de novo approach is that we're really not limited by the species that we're looking at or what isotypes. We've done work in all kinds of species and various different isotypes. And because we've been doing this for quite a long time now, we've been doing it for just over 8 years, we're able to work with a relatively small amount of sample. So we've worked with generally is at 100 micrograms, but with pure samples, we're down to about as low as 50 micrograms upon request. And we can work with samples that are less purity than what you might typically see, and our team's gotten great at doing contaminant removal as well. So now that I've gone over the background of the technology, we can dive into where does this actually fit. And as Dr. DeLuca pointed out, a couple of benefits that sequencing and recombinant expression can provide. The ability to, improve reproducibility, decrease reagent costs, and then secure the availability of these reagents really indefinitely. So these ubiquitous benefits for really anybody using antibodies for key aspects, such as in vitro diagnostics, or people that are worried about having hybridoma loss. So that's great, but at the same time, she also highlighted how we can take these sequences and take them one step further to create new reagents and tools. So whether that's doing some antibody engineering and pretty much engineering an antibody to have a different preferred FC region, or if it's generating a unique scFv, as she had mentioned, to maybe create a proof of concept construct for a novel cell therapy. For all of these things, having the sequence is really crucial. And another benefit that Dr. DeLuca had mentioned was actually having, the ability to move to recombinant systems can also have nice ethical changes as we can move away from using antibodies or antibodies generated from animals. So that leads into sort of the next topic I'd like to cover, which is what about polyclonal sequencing? So our team has become world leaders in de novo protein sequencing, and though we're very proud of all the numerous monoclonal antibodies that we've sequenced, the goal was to really expand on our capabilities and be able to work with polyclonals as well. And happy to say that we were actually the first team, to our knowledge, to successfully sequence a polyclonal antibody using proteomics alone. And since that first successful project, we've launched our RePab polyclonal sequencing service, and our capabilities have really continued to grow more robust, having completed a number of projects both internally and for our clients. So in its condensed form here, the workflow looks really quite similar to our ReMab service. In actuality, it is much more complex. We do many more digests and many more mass spec runs. However, the general flow of the project is quite similar, where we take a purified antibody, perform a number of digests, run the samples on the mass spec, and then having that data sequenced using our algorithms. And ultimately, what we end up providing is a report with full-length antibody sequences of heavy and light chains, with those heavy and light chains being paired. Now, one unique aspect of this polyclonal workflow is that we can also use B-cell NGS sequencing to sort of add in an additional layer to this, and that can allow us to enrich the proteomics data that we're generating and produce more candidate sequences. So with these capabilities, we've been able to immortalize polyclonal antibodies by identifying unique monoclonals directly from these mixtures that can then recapitulate the original binding of the polyclonal. So in fact, working with one of our expression partners, we did just that when their golden goat, unfortunately, was no longer able to produce an antibody, and they wanted to move to a recombinant structure to continue offering this product. So now, one thing I'd like to point out here is when we initiate a RePab project, we never really know exactly what the outcome is going to be in terms of the complexity of the sample. So in the case of this goat, it had actually been immunized a number of times, and as a result, had a very convergent immune response against the antigen of interest. And that led to our team identifying only 2 major forms within the mixture. Now, as I mentioned, it can really vary how many different unique sequences we're getting. A typical project, we're normally seeing about 5 to 20 unique sequences, but we've also seen over 30 in others. So for both of these forms, looking here, we did get great coverage across the heavy and light chains. And so we're quite confident in the sequences and move forward with the expression. So after we had the antibodies expressed, we performed 2 separate ELISAs and found that both of these clones depicted in the black and red lines here actually recapitulated the original binding of the polyclonal, which is shown in green. So you can see this in both of the graphs. Now, interesting fact is that we found that one of these clones actually worked much better in Western blots, while the other did not. And this truly highlights one of the benefits polyclonal antibodies can provide and why it's still important to have these and why they're so commonly used within the industry. Now, converting polyclonals can be useful, but oftentimes it may not be feasible due to certain limitations, such as sample requirements, or if you don't have the funding for such a project. However, we have also utilized our repab technology to perform antibody discovery projects as well. And in these projects, we begin with the fresh immunization, either with our clients or through partners. That's followed by a two-step purification of the resulting serum, in which we first enrich the IgG population using protein A, and then we'll perform an antigen affinity purification following that. When we perform our sequencing, this will then allow us to identify the most abundant monoclonal antibodies generated against the antigen of interest directly from the immune response without relying on the B cell repertoire. And once these sequences are in hand, we can then work with partners to have them expressed, and ultimately can then perform further characterization services to identify what of these monoclonal antibodies might be ideal candidates for your required purpose or goals. Now, there's already a lot of different discovery techniques out there. So what benefits do we bring to the table with this repab discovery technique? So firstly, our approach is species agnostic in theory, and we have had projects that have been successful in rabbits, goats, canines, sheep, chicken, alpaca, and human. We do have some limitations just based on sample requirements, but overall, theoretically, we are indeed species agnostic. And if you do have a shark antibody, I already know that our repab team would love to get their hands on one of those. Second, our approach is an in vivo method, you may be able to expect lower immunogenicity risks and higher developability of candidate mAbs. Third, if needed, we can actually do some selection for desired function prior to the actual sequencing. And finally, our approach can complement other existing techniques as we're able to identify some unique binders that may not be detected from B cell sequencing or other approaches, and may also more accurately reflect the native immune response. So, and I know that we're getting low on time, so I'm trying to do a bit of a speed run, but I wanted to highlight that we have recently completed a discovery workflow internally in which we were able to sequence a number of antibodies from an immunized alpaca and characterize the recombinantly expressed mAbs using both SPR and HDX, epitope mapping techniques. So we've been presenting posters at some recent conferences, and if you're interested in checking out this work, please follow up after the webinar and we can send over some more details as well. So key takeaways. Next generation protein sequencing is a powerful technology that can be utilized by researchers across various fields. Combining this sequencing with recombinant expression can provide long-term access and reproducibility of both mAbs and PABs, while also increasing reagent variability. And finally, with our repab polyclonal sequencing, it's possible to identify unique antibodies directly from the native immune response. And in addition to our sequencing, as I mentioned with the posters, we do also offer some characterization services such as SPR kinetic analysis and HDX-MS epitope mapping. For more information on these, you can go to our website or just send a quick follow-up after the webinar. And then finally, for those who don't know the Rapid Novar team, we are world leaders in protein sequencing, having been around for over 8 years and having worked with teams all over the globe. And I'd really just like to give a big thank you to all the members of the Rapid team, some of them pictured here during our last month celebration when we had just passed the 8,000 remAb protein sequencing projects completed. And with that, we can move into the Q&A session.
Great. So I'll take it over from here. Thank you so much, Dr. DeLuca and Dylan, for really great presentations. So we do have a few questions in the chat, so I'll start off here. The first one is for Dr. DeLuca. How stable or full is the GFP-linked SCFV in HeLa cells, given the SCFV has disulfide linkage and cells have reducing environment?
Yes, that's a great question. And this is probably why we don't have such great success with it, is that the stability. So what we're trying to do is, I mean, that's our goal right now, is to find scaffolds and CDR combinations that are more stable. And there are ways we can do that kind of rationally. At first, we just started kind of throwing things together, and that wasn't working so great. And so we've been using scaffolds that we know are stable. And then I think by doing the AlphaFold predictions, we think that might help a little bit. But I guess the question is, Are they stable? Not so much. Some are, but some are not. And that's the problem. We have to try to figure out how to increase the stability for those scFvs. And that's just our guess, is that's why we're not getting a great success rate. Yeah.
Great. All right, next one is also for, follow up to that one. Did you try mutating the cis?
Yes, we haven't yet, but we're working with Tim Stasevich down the hall, who also is invested in trying to generate these stable scFvs to track. He's tracking acetylation during transcription, and so we've been working together on that approach, but we haven't gotten very far yet. But it's on the radar for sure.
Great. Next question, also with regards to the scFvs. When building scFvs, did you try different domain orders? Are you using a G4S3 linker or another linker format?
So so a couple of things. For the linker, yes, we're using our own. We've tried several different of our own linkers. So basically, we've taken the native scFvs that worked, and we're kind of modeling our next ones off of that. And so we have a short linker that we just designed in the lab that we found worked best. It doesn't have a name or anything. It's just a linker. And to be honest, I probably couldn't tell you the amino acid composition right now, but we do have, whoever's asking, I'm happy to share that information. If I can get I just need to look up the sequence. But I will say that I think this is related. All right. So if you look at Rapid Novar, you get an annotated sequence, and it has the CDRs, the hypervariable domains, everything is mapped out and annotated. If you get the sequence from another company, the same exact antibody, those annotations are quite different. So it's not an exact science of identifying the CDRs, as Dylan probably knows this. I'm not sure if this is part of your wheelhouse, but they can be wildly different. So I wish I actually had a slide in here. I thought I was going to go too long. So if you actually compare maybe a dozen different algorithms for picking out the CDRs, they can be so wildly different. Some don't even overlap. So in terms of designing the scFv, we have a very strong feeling that identifying those CDRs is going to be a big key to it. And so that's why the AlphaFold predictions have come in. And so basically what we want to do is take predict the structure of the full bivalent folded antibody, and then predict the structure of our scFvs generated through these different combinations. And then we overlay them to try to match the structure that the scFv folds into to the native antibody, right? So the fingers that kind of reach up to the epitope, we're trying to match those to the original antibody. And so I don't know if that was part of the question, but that's another way that we're trying to optimize the scFv design.
All right. Next couple questions are for Dylan. First one, as far as antibody sequencing goes, what safeguards do you have for making sure longer VH CDR3 regions are correctly sequenced?
Yeah, sure. So I think this is probably in relation to bovine antibodies. And in terms of, how we go about the actual sequencing, because we're relying on peptides and looking at that overlap for confidence, we're able to be really confident in the sequence itself. In terms of having that actually annotated, I think that goes back a little bit to what Dr. DeLuca was mentioning about making sure that the correct annotation is implemented. Unfortunately, I don't have the full background on how to choose which annotation would be best, but knowing that it would be an antibody that you might expect a long CDR3 in, that's something that we could definitely look at, and I'm sure our bioinformaticians would be able to provide a little bit more insight than myself. So I can make a note of that and follow up after as well.
Great. And the next one also to Dylan is what would be the max number of species in a PAB sample for 100% success of full coverage sequencing? And how is the pricing?
Okay, so it's really difficult to know the max number of species in a PAB sample. So we've seen ranges anywhere from 2 to, I believe, 40 plus sequences. generally we're seeing about 5 to 20, and it's going to depend on really what you're looking for as a successful antibody. Is it going to be an antibody, you just need a couple of antibodies that bind really well, or are you just looking for as many kicks at the can as possible? And then the second part was pricing of that. So generally the projects start at about $50,000 US, and they can go up from there, or depending on what it is exactly you need completed, can vary on that front as well. But if you have a specific project, happy to set up a meeting with our team so we can go over everything and make sure that we be able to sort of meet the goals of your experiments.
Right. We got quite a few more questions here, so I'll try to get through into these as well. One it's with regards for Dr. DeLuca with regards to aneuploidy. So I guess it's a bit fragmented, this question, but with regards to aneuploidy, one, what is the frequency, and two, is it more recurrent in certain cells than others? Oh, you're on mute. You're on mute. Oh, Dr. DeLuca, you're on mute.
Okay, sorry about that. So in terms of aneuploidy in healthy primary cells, it can occur at a rate of, I would say 0.1% of the cells that are dividing will missegregate a chromosome. This increases significantly with cancer cells. So if we look at even just, we studied lots of different types of cancer cells, glioblastoma. So for example, our glioblastoma cell lines, our patient tumor samples, I would say at least maybe in the order of 5 to 8% of the cells that are dividing a missegregated chromosome that we can detect. Many of these cells die, but many of these cells can still survive and propagate. So it totally depends on whether it's a primary cell, a cancer cell, what type of cancer cell. If we are looking at cells where the aneuploidy rate is on a single division or a single division cycles is extremely high, that actually is not usually conducive for the cell population to continue to proliferate, and they die out. But it becomes more dangerous, obviously, when the aneuploidy rate is low, because those cells can thrive and divide, but yet they've been developing aneuploidies along the way. Hopefully that answers that.
Okay. This one might be a bit of a meaty question, but in terms of commercial mAbs you sequence, some companies have begun prohibiting reverse engineering of their antibodies. Do you consider this an ethical dilemma?
Was this for me?
Yes.
Can you repeat the question?
Sure. In terms of the commercial mAbs you sequenced, some companies have begun prohibiting reverse engineering of their antibodies. Do you consider this an ethical dilemma?
Yes, I do. So before we published our paper, we made sure to contact all the legal teams, all the places where we got the original antibodies. It's obviously not an issue with a hybridoma line where you own it. So for the companies where we order the antibody and then sequence them through Novar, we discuss the terms and conditions. And if most of the companies have a clause that say, you can't reverse engineer it, you can't do anything with it for commercial purposes. Most antibody companies have if you're for research purposes only, they have no problems with it. So I couldn't go and, so if I wanted to just, this isn't my, if you haven't noticed, that's not my goal to sell products. We want to give, we want, antibodies to be widely available to people. But if I wanted to, I certainly could not market the HEC1 antibody or the antibodies that we purchased first from a company. We could certainly do that with our hybridoma cell lines, or even the freely available reverse engineered, for example, our tubulin antibody, which is a gorgeous antibody, that we could, if we wanted to market it, we could. We don't. Is that kind of what, do you think that was what the question was asking?
Yep. Yeah.
Okay.
It's particularly with regards to the intellectual property and those clauses as well.
Absolutely. Yep. So we had to, I actually talked to our legal team at CSU to make sure I wasn't going to get in trouble for doing this. And as long as we're not going to sell the ones, then we're okay. Yeah.
Great. Okay. few more streaming in, so I'll try to get this one through. So another one for Dr. DeLuca. During the development phase, are there any tricks to consider when trying to get monoclonal antibodies that work well for FACS or ELISA assays?
So is the question specifically asking how we optimize the development so they will work for those approaches?
I think so. So during the development phase, are there any tricks to consider when trying to get the antibodies that work well? So I guess as you're developing or trying to figure. Okay.
Yeah, I mean, in general, just tricks to get them to work well. I have to say that we were, I mean, I don't want, knock on wood, I guess, but we were very lucky. We are not antibody development people. We are mitosis kinetochore people. So, it was very straightforward. We didn't have to do a lot of troubleshooting with just the straight taking the sequence and turning it into a recombinant antibody. That was very straightforward. Again, like I've talked to Dylan about this, we've had 100% success rate with all the sequences you give us. They all work. So there's been no troubleshooting. And we found the same thing with our hybridoma cell lines. We take the sequence, clone them into vectors, express them. And, I mentioned before, there's variability in yields, and we still don't know why. Certain antibodies we treat the same exact way. Some will yield 100 micrograms and some will yield over 2 milligrams. The quality of the antibody is never different. It's not the quality of the antibody that changes, it's just the yield. And we don't know yet why, and we could work on optimizing that. But in terms of troubleshooting from the beginning, just to get a competent, successful recombinant full-length bivalent antibody, there's really honestly not much to it. The troubleshooting really comes in with trying to make the fragments and to stabilize the fragments, if that's what they're asking. Yeah.
And so we're almost at time. I'll field 2 more questions, and then if there's anything else, please follow up with Dylan after the webinar. So next question is for Dr. DeLuca. By transfecting both plasmid for heavy chain and light chain, what was the format for the recombinant antibody obtained? You're on mute again.
So can you repeat the question? So one more time?
Yep. By transfecting both plasmids for heavy chain and light chain, what was the format of the recombinant antibody obtained?
The format of the antibody. So,
yeah.
So it folds. So the heavy chains, the light chains, you express them in the cells. In the cell, they fold correctly. They're secreted out. And so the format that is secreted into the media is a full bivalent, full-length antibody. Yeah.
Okay. And last one is for Dylan. How often do researchers and companies go from immunized animals directly to repab polyclonal antibodies and to avoid the hybridoma step?
I think more and more researchers and companies are starting to move in that direction. We've heard a lot of stories of people that have great seroresponse after their immunization and aren't able to get that from any of the hybridomas that end up going through successfully for fusion and expansion. So I would say more and more are moving towards that. We do have some people that love going right into repap. We also have people that use it in parallel with other techniques. So I think both are great options. And as I mentioned, you can get unique insights from repap itself, but there's always possibility for using multiple techniques to get the best answer for whatever your goal is.
Great, thank you. So there were a few last questions that we couldn't get to due to time. So I encourage you, if you didn't get your question answered, to follow up with Dylan, and we might be able to connect you with Dr. DeLuca to be able to answer that in further detail. But thank you again for everyone to join on today's webinar, and we will end it here. So thank you very much and have a great day.